All right, so hopefully this actually falls really nicely after the last talk, because we're also going to be looking at the effects of repeated mRNA vaccination in a way, but from the CD4 T cell side of things. And so we have the sciencey title of the talk, of course, but I think really we want to ask the question of, what do our CD4 T cells remember? And perhaps more interestingly, how much does it matter, and how does that impact booster vaccination? So I don't have to give a lot of the background. Actually, it's been covered really nicely over the last couple of days. But in terms of influenza and SARS-CoV-2 respiratory viruses, this is the messaging that we're now getting at this point in time, right? The Australian government is saying, hey, it's safe to get your flu vaccination and your COVID-19 booster vaccination at the same time. Protect yourself against both of these viruses. And we've had a particularly robust flu season this year in Australia. So we're no longer talking about primary vaccination for SARS-CoV-2. We have two cases where we have antigenically evolving viruses, where we're seeking to continually sort of update our immune response, and particularly from the antibody side of things, trying to update our neutralizing antibody responses to react to the most recent circulating variants, or hopefully variants that might circulate in the future. And again, I mentioned I don't really need to go through a lot of this sort of immunology background, but we're quite interested in CD4 T cells and their ability to provide help to B cells and support antibody responses. And so we've done a lot of work over the course of the pandemic studying primary vaccination or primary infection responses. And so we always sort of give this introduction and say, vaccine antigens make it to your draining lymph node, they initiate this germinal center response, and the output of that GC response is different types of plasma cells and memory B cells that support your long-term serological immunity. Now, CD4 T cells play a critical role in this process, of course. We've heard a number of talks over the past 2 days about CD4 TFH. So when naive CD4 T cells are primed in the lymph node, they can make early fate decisions. And some of them will start to upregulate CXCR5 and differentiate towards this T follicular helper cell phenotype. And we'll come back to that fate decision in a couple of minutes. But those pre-TFH cells, if they find a B cell that's presenting their peptide MHC complex, can then further differentiate into germinal center TFH cells, where they support BCR mutation, B cell maturation, and production of all those B cell subsets that I just mentioned. Now, that may be what happens during primary vaccination, but then it comes around to what's happening now when we continue to give these booster vaccines. We think that during booster vaccination, of course, we're predominantly recalling the memory B cell population that's circulating within the blood and that is poised to rapidly respond and increase antibody titers after a booster vaccine. But what does that mean? What are we actually doing within the lymph node? Are we initiating a new GC response? And how do CD4 T cells come into play during this immune response? And we've had some clues over the last few years from studies in human vaccine cohorts where they collected fine needle biopsies of the vaccine draining lymph nodes. And this was a study of the inactivated unadjuvanted influenza vaccine, where they found that in about half of the participants in the trial, they could see evidence of an antigen-specific or flu-specific GC response, but in the other half of participants, they couldn't. And in those that did develop a GC response, it took about 2 weeks for those antigen-specific or HA-specific GCs to develop, and then that response continued to mature over the next couple of months. But we know that when we give you an influenza vaccine, we see this rapid recall and rapid boost in serum antibody titers towards HA that occur within 6 or 7 days post-vaccination. So that initial antibody recall response is almost certainly being driven by GC-independent mechanisms. And then potentially there's contribution of GC-dependent activity later on in the response. So what does this mean from the CD4 T cell side of things, and what can we learn about CD4 T cell recall? Well, of course, we know that lymph node biopsies, although wonderful, are not necessarily feasible in a lot of our clinical trials and cohort studies. So human T cell immunologists typically focus on our circulating TFH subset, which we usually define as CD4 T cells that express CXCR5, either on their own or in combination with PD-1. And we think that these cells are a biomarker of the GC-TFH response. There's been a lot of really lovely transcriptional work showing that these cells are more related to GC-TFH populations than the rest of the CD4 memory population. And we think that tracking these cells usually gives us some insight into the biogenesis or the recall of a vaccine response. And so there's been a lot of literature over the last probably 10 years or so looking at CTFH responses to flu vaccination. And we think we understand pretty well what happens here. About a week after you get an unadjuvanted flu vaccine, you see a population of activated CTFH that appear in the blood. And these typically express ICOS or ICOS in combination with CD38. And we know that these contain HA or antigen-specific cells. And the magnitude of that CTFH response correlates very nicely with the magnitude of the antibody response to the flu vaccine at day 28 or a month post-vaccination. So that all seems nice and simple. CTFH are a great biomarker of the flu response. But what about for SARS-CoV-2? Do we see the same sort of relationship? Early on in the pandemic, there were a number of groups that looked at both primary infection as well as primary vaccination, and we showed using class 2 tetramers that there are CTFH that are elicited by primary infection and vaccination, and again, this correlates with antibody responses. But what we noticed was that over time, we started to see fewer and fewer CTFH cells when we looked at the antigen-specific population. This was some data from a study that we did looking at third mRNA dose vaccinees with, again, a spike-specific class 2 tetramer looking for CXCR5 expression. And then this was also shown recently in a paper in Cell Reports that looked similarly to what we just heard after dose 2, 3, or 4 of mRNA vaccination, and found that after each repeated vaccination, the proportion of antigen-specific CTFH seemed to make up a smaller and smaller proportion of the response. So we're potentially seeing something very different here with the SARS-CoV-2 booster vaccines than what we see with a different booster vaccine, such as influenza. And so we really wanted to understand and compare more directly what was happening with these two CD4 T cell memory populations. So we took advantage of the fact that we had access to PBMC samples from a recent clinical trial. This was a clinical trial that was looking at the co-administration of seasonal influenza or a SARS-CoV-2 booster vaccine, and participants were randomized either to receive both vaccines in the same arm or the vaccines in opposite arms at the same time. There were 56 participants overall. They received the 2023 seasonal influenza vaccine, which was unadjuvanted, and they received the COVID-19 vaccine that was available in Australia at the time, which was an XBB monovalent vaccine. And there was no primary outcome from the clinical trial, no significant difference between the two trial arms. So we had PBMCs that were available for further follow-up study at baseline, day 6, and day 28. And I think the important point about this cohort that makes it quite interesting is that it's a quite heavily vaccinated cohort. So almost 80% of the participants had received 3 or 4 flu vaccines annually over the last 4 years. And all of the participants had at least 3 prior COVID-19 vaccines, but 40% of them had 5 prior vaccines. So a lot of people in this cohort were receiving their 6th COVID vaccine over the course of 4 to 5 years. The primary vaccine course that was available in Australia was either a viral vector or an mRNA LNP vaccine, and again, we'll come back to that in a minute. And I just want to make the point clear that both of these vaccines were immunogenic. They both do what they're meant to do on the box. We saw significant increases in HI titers and SARS-CoV-2 neutralizing antibodies within 6 days of vaccine administration, and this increased a little bit further at day 28. So both of these vaccines elicited a good antibody response and did what they were meant to do, which allowed us to ask the question of, what was the immune response that got us to that endpoint. So we really wanted to focus on both the quantitative and the qualitative features of the CD4 response across those 2 different vaccines, taking advantage of the fact that we could do inter-participant comparisons here, looking at the impact potentially of 2 different vaccine platforms, as well as a combination of both memory responses and potentially de novo T cell responses to new epitopes within, for example, the XBB vaccine. So we did this across 2 different approaches. We had 40 participants where we had samples available for AIM assays. So we stimulated PBMCs with peptide pools covering either the 4 HAs that were in the flu vaccine or the XBB spike protein. And then we also had a subset of participants where we had HLA typing that allowed us to use MHC class 2 tetramers to look directly ex vivo at either the spike-specific or the HA-specific CD4 T cell responses. So starting with the AIM data and sort of getting a sense of this sort of bulk vaccine-specific response, in this case, we defined our AIM-positive cells based on 3 markers, including downregulation of CXCR4, which we've shown focuses sort of on that high avidity, non-bystander T cell population. And what we found was quite interesting. If we looked at the frequency of antigen-specific CXCR5 negative CD4 memory, we saw a flat line across the whole cohort for both antigens, HA on the left and spike on the right. So we see lots of pre-existing memory for both of these antigens across the whole cohort, but it was quite stable across day 6 and day 28 post-vaccination. Where things looked interesting was when we focused more on the CTFH population or our CXCR5 positive cells. What we see here with flu is what I would probably describe as a classic recall response. We saw a significant increase in HA-specific T cells at day 6, which remained high at day 28. And with SARS-CoV-2, we saw a slightly different pattern. We saw a small increase in spike-specific CTFH at day 6, but a much stronger increase at day 28, perhaps more indicative of a primary response to new epitopes rather than a recall response to conserved epitopes within spike. We further broke down the spike-specific CTFH responses within the cohort, looking first at the number of prior vaccines that they had received. And here we saw, similar to what people have seen previously in flu cohorts, where the individuals that had received fewer prior vaccines tended to have a stronger vaccine-induced CD4 T-cell TFH response, and individuals that had had those 5 or 6 prior vaccines had a more muted response. Additionally, when we broke the cohort down by whether they had originally received an mRNA vaccine or a viral vector vaccine, we saw what appeared to be sort of this long-term impact, this sort of long-term impact on the frequency of spike-specific CD4 memory. The individuals who were primed with the mRNA LNP vaccines had about 3-fold higher spike-specific CD4 T cells at all time points post-vaccination compared to the individuals that had been primed with the viral vector vaccines. Now, what was particularly striking when we looked at the data was that if we looked at the proportion of AIM-positive cells for each of these antigens, there was a strong difference in the proportion of the response that had that CTFH phenotype. So we're not directly comparing magnitude here, but rather the flavor or the quality of the T cells. And at every time point post-vaccination, we found that the HA-specific CD4s were significantly more likely to exhibit a CTFH phenotype compared to the spike-specific cells. So the question was, what does that mean? And what were we taking away from all of this data? So when we look overall at the AIM data, there were several things that were striking, I think. First was those sort of discordant kinetics in the recall and the expansion of the antigen-specific CTFH populations. And in particular, we thought, okay, it's very strange that we're seeing these substantial increases in spike-specific neutralizing antibodies, but we're not really seeing a lot of evidence for spike-specific CD4 T cell recall at day 6. Additionally, we see again this pattern that repeated vaccination seems to be associated with a smaller or slightly blunted T-cell response post-vaccination. And also this sort of intriguing observation that the HA-specific CD4 memory seemed to be more CTFH biased than the spike-specific memory. So this was really where we thought, okay, we have the opportunity now to come in with these tetramers and look at the cell phenotype more directly ex vivo and either confirm or potentially confuse these observations with a different measurement of the CD4 T cells. And so the tetramers that we used here were a combination of peptides. There were 3 different peptides derived from the spike proteins that we used in a pool, as well as 3 different peptides derived from the H1 or the H3 HA antigens, all of which had been conserved across multiple years of the seasonal flu vaccine. So we're quite confident that these are looking at memory recall responses when we look at these epitope-specific cells. And so using our tetramers, we can very nicely see in some individuals at baseline and day 6 post-vaccination exactly what we would expect, this very clear expansion of antigen-specific CD4 T cells, in this case showing the spike tetramer data. But we also had a lot of participants where the situation looked a little bit more like this, these stable frequencies of cells that didn't really change much pre- and post-vaccination. And when we quantified that, we can see there is a slight increase in tetramer-specific CD4 T cell frequencies at day 6 post-vaccination, again, consistent with what we would expect for a memory recall response, but it wasn't particularly striking in terms of its frequency. And this is where we were able to go back and compare some data that we had previously generated using these same tetramers in other cohorts. And so similar to what we just saw for the CD8 T cells, if we look at the fold change in tetramer frequency in convalescent individuals who received 1 dose of an mRNA vaccine, individuals who had received 3 doses of an mRNA vaccine, or this cohort where we had at least 4 doses of mRNA vaccines, we can see this progressive blunting again, where we don't see a massive amount of proliferation or expansion of antigen-specific cells post-vaccination. We do know, however, that these people are responding and are seeing spike antigen. So if we look at the classic ICOS and CD38 activation markers on our tetramer-positive population, we can see that basically everyone has a vaccine-specific response at day 6. However, the frequency and the proportion of these cells that are actually activated, again, is really variable across the cohort. And this is also something that seems to be changing over time. So if we go back to our other cohorts, again, in individuals who had received 1 or 3 mRNA vaccines, we would typically expect to see that 95 to 100% of their spike-specific CD4 cells showed evidence of activation post-vaccination. But what we're starting to see now is potentially this sort of vaccine dose-dependent decrease where individuals on their 6th or 7th mRNA vaccine are starting to show much lower levels of CD4 T cell activation following vaccination. And we kind of wondered if this was related to baseline antibody titers, if there was perhaps some effect on the antigen being opsonized or not necessarily as available post-vaccination. And we do see somewhat of a trend between the baseline XBB antibody binding titers and the spike-specific activation at day 6, although it's not particularly strong and not statistically significant. So it's a little bit unclear at this point in time what's driving this. And I will say it's not tetramer differentiation of the CD4 T cells. We know that much. So we wanted to also look and see whether we could see evidence of that difference in CTFH bias between our HA and spike-specific CD4 T cells. So here we're looking at the expression of CXCR5 and CD27 on our tetramer-specific cells, and we can see that indeed at the epitope-specific level, there's a much greater proportion of the spike-specific cells that do not have a CTFH phenotype compared to our HA-specific cells. And if we look at the co-expression of CXCR5 and PD1, we can also see the HA-specific cells, even at baseline, tend to co-express CXCR5 and PD1, whereas the spike-specific cells at baseline have a population instead of CXCR5 negative, PD1 positive cells. And so again, this is pre-vaccination. So this is the phenotype of these cells as they're maintained in long-term memory, suggesting that again, for some reason, there's a difference in the quality of the CD4 T cells elicited by these two vaccines. And the last piece of data that I wanted to show that I think was quite interesting was we went back and we looked just at bulk CTFH or bulk memory CD4 activation and the relationship with antibody responses. And I had said before, many studies have shown that CTFH activation at day 6 correlates nicely with day 28 influenza HI antibody titers. And again, we see that here in our cohort. But what we see is that for SARS-CoV-2 neutralizing antibodies, the correlation is actually with the activation of the memory CD4 T cell population. So again, driving home this idea that there is something qualitatively distinct about the CD4 memory in the SARS-CoV-2 vaccination context compared to HA vaccination. So overall, what we seem to find is happening, combining both the AIM data and the tetramer-specific data, was that at baseline, we see some differences in how HA and spike-specific CD4 memory is maintained. At day 6, for the HA-specific CD4s, we see proliferation of the CTFH population and activation of both the CTFH and the memory CD4 populations. In contrast, for spike, we see activation, again, both of these populations, but minimal proliferation, particularly in individuals who are highly vaccinated. And then by day 28, we see resolution of the acute activation of all the HA-specific CD4 memory, and it's not really clear if there's much of a de novo CD4 response at that time point. However, for spike, we do start to see increases now in TFH that we suspect are probably primary TFH responses rather than the recall that we saw at day 6. But the point, and I think the take-home message that's really interesting to me at least, is that all of these responses appear to support antibodies. Again, we saw that both vaccines were immunogenic. We didn't see any sort of non-response to the SARS-CoV-2 vaccine in terms of neutralizing antibody responses. So there do seem to be pathways for these different CD4 qualities to still be influencing and supporting a productive antibody response. And so with that, really the take-home messages are exactly that. The CD4s are correlating for both of these vaccines, but we really want to try to understand why we see these differences in CTFH biases across antigen specificity. And again, also trying to understand the basis of this observation that really the sort of repeated vaccination seems to be associated with a more stable CD4 T cell pool or population. And so with that, I'd just like to thank the individuals in the lab, particularly Palak and Kathleen, who were fantastic in generating a lot of this data, our trial participants and funders, and I'm happy to take any questions.